Good day everyone. Today we will discuss about membrane lipids and membrane fluidity. The physical state of the lipid of a membrane is described by its fluidity or viscosity. So consider a simple artificial bilayer compressed of phosphatidylcholine and phosphatidyltretanolamine whose fatty acids are largely unsaturated. If the temperature of the bilayer is kept relatively warm, the lipids exist in a relatively fluid state. Above the transition temperature, the lipid molecules and their hydrophobic tails are free to move in a certain direction, even though they retain a considerable degree of order. At this temperature, the lipid bilayer is best described as a two-dimensional liquid crystals. As in crystals, the molecules still remain a specified orientation. The long axes of the molecule tend toward a parallel arrangement, yet individual phospholipids can rotate around their axis or move literally within the plane bilayer. If the temperature is slowly lowered, a point is reached where the bilayer distinctly changes. Below the transition temperature, the movement of the molecules is greatly restricted and the entire bilayer can be described as a crystalline gel. The lipid is converted from a liquid crystalline phase to a frozen crystalline gel in which the movement of the phospholipid fatty acid chain is greatly restricted. The temperature at which this change occurs is called the transition temperature. So always remember that the phospholipids with saturated chains pack together more tightly than those containing unsaturated chain. The greater the degree of unsaturation of the fatty acid of the bilayer, the lower the temperature before the bilayer gels. Another factor that influences bilayer fluidity is the fatty acid chain length. The shorter the fatty acid chains as a phospholipid, the lower its melting point. The physical state of the membrane is also affected by cholesterol. Because of their orientation within the bilayer, cholesterol molecules disrupt the close packing of fatty acid chains and interfere with their mobility. The presence of cholesterol tends to abolish sharp transition temperatures and creates a condition of intermediate fluidity. Now, let's move to the importance of membrane fluidity. What effect does the physical state of the lipid bilayer have on the biological properties of the membrane? Membrane fluidity provides a perfect compromise between a rugged ordered structure in which mobility would be absent and a completely fluid, non-viscous liquid in which the component of the membrane could not be oriented and structural organization and mechanical support would be lacking. In addition, fluidity allows two interactions to take place within the membrane. For example, membrane fluidity makes it possible for a cluster of membrane proteins to assemble at particular sites within the membrane and form a specialized structure, such as intercellular junction, like capturing photosynthetic complexes and synapses. Because of the membrane fluidity, molecules that interact can come together, carry out necessary action, and move apart. Membranes arise only from pre-existing membranes, and their growth is accomplished by the insertion of lipid and protein components into fluid matrix of the membranous sheath. Many of the most basic cellular processes, including cell movement, cell growth, cell division, formation of intercellular junction, secretion, and endocytosis, depend on the movement of membrane components. The internal temperature of the most organisms fluctuates with the temperature of the external environment, since it is essential for many activities that the membrane of a cell remain in a fluid state. Cells respond to changing conditions by altering the types of phospholipid of which they are made. Maintenance of membrane fluidity is an example of homeostasis at a cellular level that can be demonstrated in various ways. For example, if the temperature of a culture of cells is lowered, the cells respond metabolically. The initial emergency response is mediated by enzymes that remodel membranes, making the cell more old resistant. Remodeling is accomplished by first is desaturating single bonds in fatty acid chains to form double bonds. Desaturase is the desaturation of single bonds to form double bonds that is catalyzed by enzymes. The second one is reshuffling the chains between different phospholipid molecules to produce ones that contain two unsaturated fatty acids. So it greatly lowers the melting temperature of the bilayer. Reshuffling is accomplished by phospholipases, which splits the fatty acid from the glycerol backbone, and acetyltransferases, which transfers them to different phospholipid. In addition, the cell changes the types of phospholipids being synthesized in favor of ones containing more unsaturated fatty acids. As a result of the activities of these various enzymes, the physical properties of the cell membranes are matched to the prevailing environmental condition. Maintenance of fluid membranes by adjustments in fatty acid composition has been demonstrated in a variety of organisms, including hibernating mammals, pond-dwelling fishes whose body temperature changes markedly from day to night, cold-resistant plants, and bacteria living in hot water springs. The lipid bilayer consists of two distinct leaflets that have a distinctly different lipid composition. One line of experiment that has led to this conclusion takes advantage of the fact that lipid digesting enzymes cannot penetrate the plasma membrane and consequently are only able to digest lipids that reside in the outer leaflet of bilayer. As shown on the figure, if intact human red blood cells are treated with lipid digesting phospholipase, approximately 80% of the phosphatidylcholine, which is the PC, of the membrane is hydrolyzed, but only about 20% of membrane's phosphatidylethanolamine, which is the PE, and less than 10% of its phosphatidylserine, which is the PS, are attacked. This data indicate that compared to the inner leaflet, the outer leaflet has relatively high concentration of PC and low concentration of PE and PS. The biological role of lipid asymmetry is not well understood except in a few cases. All of the glycolipids are in the outer leaflet where they probably serve as receptor for extracellular ligands. Phosphatidylserine, which is concentrated in the inner leaflet, has a net negative charge of physiologic pH, which makes it a candidate for binding positively charged lysine and arginine residua, such as those adjacent to the membrane-spanning helix of glycopyrene A. We have assumed in the previous discussion that lipid molecules are uniformly distributed with each leaflet of the bilayer, and it's only the proteins that make the membrane unsafe. Actually, a large body of evidence gathered in a recent year suggests that the outer leaflet of the plasma membrane of many cells contains specialized regions of microdomains that possesses a distinctive lipid composition. In 1980s, extraction of cultured cells with cold, non-ionic detergents such as Triton X100 left behind a considerable fraction of the membrane lipids. Further analysis indicate that membrane residua left behind after the initial detergent extraction consisted primarily of cholesterol and lipids with long saturated fatty acid tails, particularly sphingolipids. When studied in artificial bilayers, cholesterol and sphingolipids tend to pack together tightly to form microdomains that are more regulated and highly ordered than surrounding regions consisting primarily of phosphoglycerides. Because of their distinctive physical properties, cholesterol-rich microdomains tend to float within the more fluid and disordered environment of the artificial bilayer. As a result, these patches of cholesterol and sphingolipid are referred to as lipid drops. When added to these artificial bilayers, certain proteins tend to become concentrated in the lipid drops, whereas others tend to remain outside their boundaries. GPI-anchored proteins show a particular fondness for the outer regions of the bilayer, as seen in this figure. The existence of cholesterol-rich lipid drops in the plasma membranes of living cells has been a major source of controversy in the field of membrane biology. So recent studies support the existence of lipid drops but suggest they are small and undergo continual formation and dispersal. Lipid drops are thought to be more than structural compartments within plasma membrane. They are postulated to serve as floating platforms that concentrate particular proteins, thereby organizing the membrane into functional components, as what is seen in the illustration. For example, lipid drops are thought to provide the favorable local environment for cell surface receptors to interact with other membrane proteins that transmit signals from the extracellular space to the cell interior. So it has also been speculated that two of the proteins involved in Alzheimer's disease, amyloid precursor protein and gamma secretase, might be preferentially concentrated in lipid drops. If this is the case, it might explain why cholesterol-lowering drugs decrease the risk of developing ED. Lowered cholesterol levels will decrease RAP formation, which in turn would likely decrease the production of the disease-producing amyloid beta peptide.